Welcome back to my channel, The Life in the Womb. Those who have not subscribed yet, please click the subscribe button given below so that you will be getting updates in future. In this session, I would like to take something about gametogenesis. And gametogenesis I am planning to take in 3 sessions. The first session we will be having a quick recap about mitosis and meiosis. The second session we will be covering the spermatogenesis, and the third one we will be covering oogenesis. So before going on to the gametogenesis proper, let's see from which cell these gametes are getting developed. So gametogenesis, as we all know, it is the process by which the gametes are formed. So these are formed from the primordial germ cells which are seen during the embryonic period. You call it as primordial germ cells. These primordial germ cells will be giving rise to the future gametes. So where will you see these primordial germ cells? During the second week of an intrauterine period, you have a bilaminar germ disc formed with epiblast and hypoblast. The details of it, you can go and see the session under the development of second week of intrauterine period. In that session, I have given the details of the epiblast and hypoblast. So in the bilaminar germ disc, you have the epiblast and you have the hypoblast. So the primordial germ cells are first seen in the epiblast. In this region, you have the primordial germ cells seen during the second week of intrauterine period. Later, what happens to these primordial germ cells? They will be migrating from these epiblast into the developing yolk sac. In the second week itself, you can see the yolk sac here, and during the third week, by the end of the third week, you will get the folding of the embryo and all, and you will get the yolk sac like this. So the primordial germ cells which are seen in the epiblast will shift from this position to the yolk sac by the end of third week of intrauterine period. Now later what happens? It should anyway reach the developing gonads. So in order to reach the developing gonads, the cells will again start migrating through the dorsal mesentery into the posterior abdominal wall, where you get the developing gonads. And there it starts developing. In case of males, you call the process known as spermatogenesis, and in case of females, you call the process as oogenesis. So the primordial germ cell is actually having 2 sets of haploid chromosomes, that is, it is diploid in number. So what is actually happening during gametogenesis? There should be some reduction division happening for this diploid cells, because during fertilization we should be able to restore the number of chromosomes. So before fertilization, the 2 sets of gametes should be having just the half number of chromosomes. Then only after fertilization you will get the exact number of chromosomes. So there are mainly 2 processes happening for the primordial germ cells which are there in the developing gonads. So the primordial germ cells in the developing gonads undergo 2 types of division. One is gametogenesis. In case of males, you call it as spermatogenesis, and in case of females, you call it as oogenesis. So what is happening during gametogenesis? In gametogenesis, the highlight is the meiotic division, where the chromosomes are made to half the number of chromosomes, so that the diploid number is actually made into haploid. And what is the next process? Next process is known as cytodifferentiation. So cytodifferentiation is a process by which the gametes are actually becoming mature enough for the fertilization process. So the details of the gametogenesis I will be dealing in the next session. In order to understand gametogenesis, you should have a thorough knowledge about mitosis and meiosis. Of course, mitosis and meiosis you are thorough during your school days. This is just a recap of the same, so that especially in oogenesis, you will be coming across at birth, the female child is having oocytes at the arrested phase of prophase, and after ovulation, it is having the oocytes at the arrested phase of metaphase. So such terminologies you will be coming across when we deal with the gametogenesis. So in order to understand those concepts, you should know what do you mean by mitosis and what do you mean by meiosis. So let's see the mitosis first. So in mitosis, before moving on to the major divisions, there is a phase known as interphase. So interphase means it includes G1, S and G2. G1 means gap 1, S means synthetic phase and G2 means gap 2. So it is during the synthetic phase where the DNA is duplicated. So mitosis is a process by which the maternal cell will be actually duplicating itself and will be equally dividing the number of chromosomes to its daughter cells. So if you take a daughter cell and if you take a maternal cell, both will be the same with the same number of chromosomes. That's the peculiarity of mitotic division. So let's see what is happening. So if you take a cell, usually the nucleus will be containing DNA just in a crumpled pattern. So what is happening during the first phase of mitosis? You call it as prophase. So during prophase, the DNA are actually neatly arranged in the form of chromosomes. And after duplication, you have a centromere with the same chromosome split into two, and you call those as sister chromatids. So in the prophase, you can see the chromosomes very beautifully arranged inside the nucleus, and you are also not seeing the nuclear membrane at this phase. There will be again one more structure formed simultaneously during prophase. They are known as mitotic spindles. Why you need the mitotic spindles? So mitotic spindles are just thread-like structures which you need to pull the chromosomes apart. That is the need for the mitotic spindles. Now we are moving on to the next step, that is metaphase. So you have to keep one point in mind. The chromosome number is still diploid, 2n. In metaphase, now the chromosomes are getting ready. So the chromosomes are aligned in the center, and you have the mitotic spindles attached to all those sister chromatids so that they are just getting ready to pull the chromatids to the ends of the cell. And what is happening during the next step? The next step is known as anaphase. So from metaphase, when you move on to the anaphase, you can see that the mitotic spindles have already pulled the sister chromatids to the two poles of the cell. And there you can see the formation of nuclear membrane around the sister chromatids. So you can see there are two centromeres here. As they are pulled apart, the centromere is also getting equally divided, and again you will be getting two centromeres at either poles, and they are actually getting covered with the nuclear membrane. Now what happens in telophase? So telophase is the next phase where the nuclear membrane is very beautifully formed, but this is actually again within the same cell. Now it's time for the cell also to divide into two equal halves. That is what is happening during the process known as cytokinesis. So during cytokinesis, there is actually the splitting up of cells into two equal halves, and the nucleus will be going along with each cell. So let's have a look. There are two centromeres with the chromosomes. So you call it as 2n. So this genetic makeup is same as that of the maternal cell. So that is what is happening during mitosis. So the chromosomes are just getting duplicated during the synthetic phase, and they are getting equally divided. So in the beginning and at the end of the mitotic division, the number of chromosomes present in the cell is just the diploid number. And now what is happening during meiosis? Meiosis division usually happens in human beings during sexual reproduction. So let's see what is happening during meiosis. So meiosis is not a single step as you have seen in mitosis. Meiosis happens with two phases. That is, you will be having a meiosis 1 followed by meiosis 2. So let's see what is happening during meiosis 1 and meiosis 2. Almost the steps resemble the mitosis. Prophase, metaphase, anaphase and telophase. You will be having all those steps. But let's see what is happening in meiosis 1 compared to mitosis. So let's see the first step. So in meiosis 1, in prophase, what is happening? You have the diploid number of chromosomes, same as that of the mitosis. And in prophase, you can see the chromosomes beautifully arranged. And towards the end of the prophase, you can see that a part of DNA is being exchanged between the homologous chromosomes. So you can see that a bit of it is gone and attached to the other set, and a bit of it from the other set is getting attached to this side. So there is actually exchange of DNA material. And what is happening during the next phase? Metaphase. So in order to specify the prophase and metaphase of meiosis 1, you can call it as prophase 1, then you can call it as metaphase 1. So same as that of the mitosis, you have the metaphase where the chromosomes are arranged in the middle and you have the mitotic spindles. So you can see that the mitotic spindles are attached only to one set, whereas in the mitosis you have this chromosomes arranged in the center and the mitotic spindles are attached to each set so that it is equally divided. But here if you take one set of chromosome, you have one set of mitotic spindles, and to the other set of chromosome you have the other set of mitotic spindle. So let's see what is going to happen. What do you get? The next step. From metaphase 1 you have the anaphase 1. Anaphase 1, you can see that the entire chromosome set is actually moving on to either poles. So it is not getting divided at the centromere. That is the main point. So what happens? If you consider this as N and this as N, here also you have this as N and this as N. When during anaphase 1, you have the chromosome set going on to either pole. So you have N here and you have N here, and in anaphase you have the nuclear membrane being formed. And what will be happening during telophase 1? During telophase 1, the nuclear membrane will be well formed. So again you will be having N and N. Still the cell is seen as a single cell. So still the number of chromosomes, you can call it as diploid. After that, that is during cytokinesis. Cytokinesis, what happens is each cell will be moving apart. Each cell will be dividing and moving apart, and they will be having only this single set of chromosome. That is, they will be having only N number of chromosomes. So what has happened? We started with 2N. After prophase, metaphase, anaphase and telophase, what you have is 2 cells with N number of chromosomes. But still you have the centromere and the chromosome is paired. That means you have 2 sister chromatids. So that is what is happening at the end of meiosis 1. So at the end of meiosis 1, the cell which was diploid is made into haploid but with paired chromosome. That means there are 2 sister chromatids for each pair. So what is the difference between mitosis 1 and meiosis 1? In mitosis, what is happening is the centromere is getting divided. So there is equal number of chromosomes in the daughter cells as well. So they started with 2N and they ended up with 2N, but you have the unpaired set of chromosomes towards the end of mitosis. But in meiosis, what happens is they started with diploid number, but at the end they are having just haploid number of paired chromosomes. Now it's time for meiosis 2 division. So meiosis 2 division, it is just like the mitosis division, because now there is no more reduction in the number of chromosomes because the chromosomes are already made into N. Now what is happening is just equal to mitosis division. So here you have N number of chromosomes with a centromere and 2 sister chromatids. Now what is happening is, so this is prophase where the chromosomes are beautifully arranged. Then you have the metaphase. So since these reactions belong to meiosis 2, you can call it as prophase 2, then followed by metaphase 2. So what is happening during metaphase 2? You can see the chromosome is arranged in the center. Now you have the mitotic spindles. They are pulling the same set of chromosomes from either side. So that means now it's time for the centromere to divide. So it is almost resembling the mitotic division. Now what happens after metaphase? It will move on to the next phase known as anaphase. So anaphase is a process by which the chromosomes are pushed or pulled towards the two poles and there is formation of nuclear membrane. So you can see that there is division of the centromere. But still you have the sister chromatids equally separated. So again you have N and N, which is equally divided between the two nuclei. And in telophase what happens is you get one N from the same cell and again another set 1N and 1N from the same cell. So ultimately if you take, you had one single cell with diploid number of chromosomes and towards the end of meiosis 2 you are having 4 cells. This is the first cell, this is the second, this is the third one and this is the fourth one with n number of chromosomes. So what is the difference between the daughter cells of meiosis 2 and meiosis 1? At the end of meiosis 1 you are still having haploid number of chromosomes. Again at the end of meiosis 2 you are having 4 cells with haploid number of chromosomes, but at the end of meiosis 1 you are having it paired. You had a centromere with 2 sister chromatids, but at the end of meiosis 2 you are having unpaired set of chromosomes with n number. So the unpaired set you are also having in mitosis as well. You have a single centromere with a single process. There are no 2 sister chromatids for a centromere. So the same type of event is happening in part 2 of meiosis. That is meiosis 2 almost resembles mitosis, but meiosis 1 is actually a reduction division. So it is this process which helps to divide the diploid number of chromosomes into haploid number. If this is not happening like this, what will happen? You will be getting the gametes with 2n, that is diploid set of chromosome. So what will happen after fusion? If one gamete is having 2n, so just imagine one set of gamete due to some defective meiotic division is having 2n as the byproduct of the meiosis and the another gamete. So suppose this is oocyte and this is spermatozoa. The spermatozoa is having normal n after the normal meiotic division and this oocyte is having diploid number due to some error in the meiotic division. What will be the product? The product will be having 3n, that is triploid number of chromosomes. So that is actually a chromosomal error. So all these errors can happen during at any point of mitotic division or meiotic division, and if these errors are happening during the embryonic period, the maximum number of daughter cells will be affected because it is during the embryonic period you have more and more number of cells being formed. So if these errors are happening during the intraembryonic period, what usually happens is more and more number of daughter cells are affected and that will result in major anomalies. The same way, in this case, if one set is actually staying there without moving on to the next, there will be an extra chromosome. So what you call, there will be 2n plus an extra chromosome in one of the cells with chromosomes. So what do you call that condition? Suppose if this extra set is seen in the 21st chromosome, apart from the 2n number, if one sister chromatid is not moving apart, you will be getting an extra chromosome for that single chromosome. Say for the 21st chromosome you are getting an extra chromosome. So the number will be 2n plus 1. So that condition is known as trisomy. You will be getting an extra chromosome in the 21st chromosome. So likewise you will be getting different sort of errors. So trisomy 21 is a very common condition, you call it as Down syndrome. So in order to understand all these syndromes, you should have a thorough idea about mitosis and meiosis. Hope I have cleared most of the doubts you had during mitosis and meiosis. So make it thorough, the mitotic and meiotic divisions, so that when we start dealing with gametogenesis, it will be easy for you to grasp the idea and you won't go wrong when you study gametogenesis. So this is all about Mitotic division and meiotic division in a nutshell. Please keep on watching. We will be doing the next sessions soon. Thank you.